person in charge on the course is actually Professor Ramona Sorek. This is her email right here. But for any question regarding the material or administrative or anything at all, you should first address me, but you are very welcome to address her as well. She's usually very responsive, so you can have both our mails or emails. The course book, or the book that we will use along the course, is this one, Molecular Cell Biology, by Lodish, 5th edition. It is available in the Hermann library, and I have it as a PDF file here. I can't put it on the website because of copyright issues, but you're very welcome to take it directly from me during the break or anytime you like. Okay, the syllabus. So these are the course chapters. So the three first ones are more maybe general biology. The first is biochemistry of the cell, which we will start with today. The second goes into the structure and function of proteins. The third, genetic mechanisms. Again, the three first chapters are more general biology, and we will discuss as much as possible. We'll discuss it in the context of neurobiology. And the three next chapters are gradually built to present the idea of signal transduction across or between cells. And in order to give a good basis to this concept of signal transduction, we start with learning a lot about the biomembrane, which is the wall across which the signal is transduced. And then we have more of a global chapter about transport of ions across cell membrane, and the third chapter in this group introduce a superfamily of very important receptors in nature, G-protein coupled receptors. And we then sort of go back to more basic molecular topics, such as modifications that genes undergo post-transcription, and the last chapter will be we will name a few techniques, genetic techniques that serve us in the labs. Okay, now I think that biology courses tend to be maybe less structured than courses with some strong mathematical element in them, such as chemistry and physics. But we don't like it to be just a list of anecdotes, and we do try to build it gradually. But still, nevertheless, we sometimes go back to some topic which has already been discussed, with a different perspective or a deeper point of view. Okay, the final grade is built of three components. The first is participation in paper discussion. So every once in a while you will receive a paper that you will read at home, and then we will read it together in class. So this is 20% of the final grade. And the idea here is not to push yourself into the discussion in class or to show that you know the material or anything, but to truly try to understand the paper together in class. And in order to do that, we have to understand the basic questions asked in the paper before coming to class, such as what is the research question that's being asked in the study, what are the techniques that are being used to implement or to try to answer this question, and so on. And coming with this knowledge with this information about the article, you will receive the full grade of this component. And this is most usually the case, okay? So last year's students, I think 99% of them, we didn't have 100 students, so it can't be 99%, but the great majority received the full grade from this part. And 40% are given for the final, for a written exam, which is an American-style exam, multiple choice test, which will be held in the last class of the semester. Okay, so you can put it in the diary. And the last part, again, 40%, is a home assignment which you will receive or will you perform right after the semester ends. So you will have around 2 or 3 weeks to prepare this assignment. And in this assignment, you choose some paper, any paper you like, and after you receive my approval, then you have some questions to answer about it. And again, you receive some grade and some feedback about it, and these are the last 40% of the final grade.
The last one is written.
Sorry?
The last one is a written answer to the question.
Yeah, it's written. It's not oral or anything. Yeah, it's written on the side notes. Okay, at the end of each chapter, I'll bring you a question page. This is absolutely for self-use or for your own benefit. It's not. You will not be graded on it. It's not a part of your final grade. You answer it alone at home or with your friends, and then we answer it together in class, clarifying any points that need to be clarified. Okay, and the idea is you have two. The rationale has two reasons. So the first is that the questions in this question page at the end of each chapter are taken from the same question bank as the question of the final exam. So it is written in the same style exactly, and it's at the same level of the questions of the final exam. So if you feel okay with the question pages during the semester, you should feel okay, when getting closer to the exam. And and in addition, it will serve you as a way to go over again the material of the chapter and see where you stand. Okay, this is kind of a request from last year's students. They asked for a term list, a full term list that have been discussed during the course. And okay, from one side, I think it's not a bad idea. Okay, because I think, the way I see it, one of the main goals of this course is to learn a new language, the molecular biology language. And I think maybe one of the most fascinating things, for me at least, in the studies that the ICMC has to offer you is that you hear, different people speak different scientific languages on the same topic. So, for instance, you can explain how memory is being formed by talking in a cognitive scientific language or by mathematics. It has a good explanation for how memory can be formed, and also molecular biology has some explanation. So here we will try to acquire a new language, so to speak. And so a term list makes sense in this sense. But I'd like to give you this list, if at all, maybe 3 or 4 weeks before the exam, just that you will not feel that it is some kind of a checklist that you can check as we go, because it will be, it's like too reductionist approach for the studies of this course. I think it's a shame. So I will prepare this list, but maybe later on in the semester. We still don't have a website. My sincere apology. We have been moved to another system, and I still don't have an access. I'm trying to open this access as fast as I can. And meanwhile, I will pass in a few minutes a blank sheet. Just fill in never mind the name, just put your address, your email address here, and I will, because this is the only way I will have to communicate with you in the coming days. I think that in 2 or 3 days I will open the website and I will send you the link as soon as I can. Okay, 2 more things. First, okay, so the course does not assume any prior knowledge in biology. But the learning rate is maybe a bit more faster than that should be if you will really assume no prior knowledge in biology. This is kind of, I think, the style of courses for second degree, and particularly in the ICMC. So those of you without any biological background, you might feel sometimes that we go maybe a bit too fast on the material. So okay, so first, you can always read at home any terms or words that you quote and do not know. And the 2 main sources for that would be, first, the Lodish textbook, and second, Wikipedia. Because we most of the time we do not go too deep that you cannot find the material in Wikipedia. That's the truth. And second, I'm here exactly for that, so you can ask me any questions along the course, after the class, or whenever you feel like, on email. You can meet face to face, whatever you like. But anyway, don't wait to do. Usually, I suggest that you won't wait even to the end of the class, and for sure, don't wait to the end of the semester to find out that, I don't know, maybe 50 terms second year I give this course in English, and still it's not my native language, so it's still a big challenge to me. You are very welcome to correct me during the course, during the class, and if you feel uncomfortable to correct me during the class, just do so during the break. It will be very helpful. Any native speaker? We do have some native speakers. So, this is my request for you. Okay. I'm sure it will not be the first or the last time that you do a name round this week, but for my sake, let's do it just one time. So everyone will say his name or her name, and what is his or her first degree or second degree if he has any, and from which university. So my name is Shachar, as you already know, and I did my first degree in chemistry and biology here in the Hebrew University, and then moved to the direct PhD program of the ICMC. I mean, this would be the third. This is the beginning of the third year of the PhD. So let's start with the front row.
Adar, I studied here psychology and cognition in Hebrew University.
Nina, the same, studied psychology.
Nimrod, I studied psychology and American in the humanities here in the Hebrew.
I'm Enrico, and I studied medicine in Berlin.
Enrica?
Enrico.
Down psychology at Open University. Some courses, basic courses in methodology.
Yoni, psychology, NYU.
Daniel, I studied mathematics and computer science in the Open University.
Nora, I studied computer science and cognitive science here at Hebrew University.
I'm Oren. I studied communication engineering in Be'er Sheva, in the university, and computer science here in the university.
I'm Haran. I studied in the University of Haifa, psychology, and Ofakim, humanities.
I'm Bettina, and I studied at Ulm University molecular medicine.
I'm Kate. I study neuroscience at Brigham Young University.
I'm studying biology.
Hi, Adi. I studied at Bar-Ilan University, life science, this year.
So how many ICMC students? Please raise your hand. And students from the neurobiology department? No, for now. Okay. Not for this one. Okay. So I'll pass these pages. Please fill your reading. So the first, okay, in each presentation for each chapter, you'll see the first slide. All of this material, also the basic information about the course will be uploaded, obviously, to the website the minute we will have one. And in the beginning of each presentation, I wrote the chapter from Lodish, from which the backbone of the presentation is taken. So some examples, some extensions are not included in the textbook, but again, the backbone or the great majority of the material from each presentation is taken from the chapter written in the first slide. So basic biochemistry of the cell. Well, in the level of description in which we will discuss today, there's a lot of organic chemistry in the biology. So this is where the term biochemistry enters. This is why we call it biochemistry. We will start by learning the different interactions and bonds exist in nature between atoms. We see that some of them are quite transient and others are quite stable. We will see their effect in biological systems. We will meet a general concept of chemical building blocks in the cell. So macrostructures are built from monomers, from single units that comprise bigger structures. And it's true for many levels of biology, so we will introduce this concept of building blocks. We will get to know the idea of chemical equilibrium, of chemical equations. And we will end by saying a few words about biochemical energetics. the strongest bond exists between two atoms is the covalent bond, in which a pair of electrons is being shared. So, for instance, if you take a hydrogen atom and another one, so under certain conditions, temperature and pressure and so on, each of the electrons that came originally from one of the hydrogen can be shared by the other one. So now the two electrons are actually being distributed across or around the two nuclei of the two hydrogens. And so this is what we call a sharing of a pair of electrons, and this is what stands in the base of the covalent bond. This is actually the covalent bond. Now each element or each atom in nature has a certain number of electrons in its circumference, which allow it to attain a certain number of covalent bonds with other atoms and certain geometry in space. So, for instance, oxygen would usually be found being covalently bonded with two other atoms, phosphor with 5, and maybe I can take this concept, and carbon with 4 covalent bonds usually. Now, nature is what we call, or biology is carbon-based. If you ever heard this expression, that we will soon see why it's so. But the idea is that most of biomolecules are arranged around a central carbon atom, and most of the biomolecule backbone are built of carbons. So we are most interested in carbons, and carbon can contain 4 covalent bonds. And because we like carbon the most, so I'd like to say a word about the configuration in space of 4 covalent bonds. So if you take 4 spheres linked to a central one, as in this substance methane, which has a central carbon atom and 4 hydrogen distributed symmetrically around it. So if you take 4 spheres linked to a central sphere, and you let it attain the most stable configuration in space, you'll get a tetrahedron, which is this shape. Now, okay, so here the 4 surrounding spheres are identical. They are all hydrogens. But if we take another substance, a different substance in which we have 4 different such spheres, and they all have to be different from one another, linked to a central sphere, we can have 2 different configurations in space, both of them tetrahedral, but still they will not be able to overlap in space. Thanks. And the relation between these 2 configurations is that they are mirror images of each other. The most classical example of a mirror image of 2 objects is your hand, that cannot be overlapped in space. Because this is not overlap, because they are front to front. You have to have overlap, you have to have back to front. And there is no overlap. So, and these 2 molecules are called stereoisomers. And although they look quite the same, maybe in this slide, they have distinct functions, and they can, one of them would link to a protein and give some, the output will be initiating of some cascade, and the other one would not. So they have a very distinct biological function. Now, 4 covalent bonds can also attain the configuration of a planar triangle if you allow the existence of a double bond, as in formaldehyde. So this is the shape in space of a planar triangle. Around the single bond, rotation can occur in the axis that links between the 2 atoms. In the double bond, there is no rotation around this axis, the bond axis. So it has a function in giving some rigidity to some regions of biomolecules. Okay, covalent bonds can sometimes be polar. They can have some dipoles across them. And to understand why it is so, we would introduce the notion of electronegativity. So electronegativity is the ability of an atom to attract electrons to reach the center. and if one atom has stronger electronegativity than another, it will attract or the electron crowds, or the electron spend more time around its nucleus, giving it a delta, a negative delta charge around it, and the other atom from which the electron has been taken would attain a positive delta charge, and a dipole is formed. So this is water molecules which have dipole across them, because oxygen is much more electronegative than hydrogen. And dipoles can, as the water molecules, can form electrostatic interactions with each other or with other molecules. So we now move to a family of much weaker interactions than the covalent bond, starting with ionic interaction. So ionic interaction results from attraction of a positively charged ion with a negatively one. So first of all, ion is a molecule that has an unequal number of protons and electrons, and is hence not neutral. A positive charged ion is termed cation, and a negatively one is termed anion. I'm not sure about pronunciation here, anion. And the electrostatic interaction between these two is exactly the ionic interaction. Now, pure ionic interaction cannot really exist, because if you let the ionic interaction to be formed, then you have some degree, most of the time, of electron sharing, or actually of covalent bonding. Now, there are certain characteristics for ionic bonding and for covalent bonding, and one can differentiate better between these two bonds. We will not do it here. We don't care too much about the overlap maybe between these two definitions. But covalent bonding is the pure sharing of electrons, and in ionic we have some degree of electron sharing and maybe more degree of positively charged interaction. And the most classic example of ionic interaction is when you put ions in aqueous solution. So the charged ions are not being left naked, and they are rapidly surrounded by water molecules. Forming a shell around them. So this is magnesium, which is a positively charged ion, and is held, and this is why the oxygen facing towards the magnesium with its negative delta charge, and will be the opposite if this picture was depicted for instance, for chloride, which is negatively charged. The hydrogen bond is the interaction between a partially positively charged atom, which happens a lot because hydrogen is very low in its electronegativity, so most atoms will succeed to produce a positive delta charge upon it. So it is the interaction between a positively charged hydrogen and an unpaired electron from another atom. So for instance, water molecules have hydrogen bonds between them. This hydrogen here is partially positive, we already know why, and it interacts with the unpaired electrons here from the oxygen via a hydrogen bond. A few things to know about hydrogen bonds. First is that the linear arrangement, meaning that is the strongest, and by linear I mean that the oxygen, hydrogen, oxygen are all aligned along a straight line. So if I take this molecule and give it a slight tilt, so that the OH will no longer be on a straight line with the HO, then the bond will be weakened. Another thing to know about hydrogen interaction is that it weakens the bond in the other side of the hydrogen. So this is the hydrogen bond, and because of this bond, which is a covalent bond, weakens a bit, and hence lengthens. It becomes a bit longer than would be the case if we would not have this hydrogen bond, because it sort of steals some of the energy from this bond. So if I can say it in the reverse manner as well. If I cut this hydrogen bond, then this bond becomes shorter, having more energy. Okay, so solubility is the ability of substance to be dissolved in aqueous solution. Solubility of, well, minute before, the uncharged solubility of charged molecules, which was the previous slide, is thanks to ionic interactions, as we saw in the case of ions, right, being placed in aqueous solution. And what stands in the base of stabilizing uncharged molecules in aqueous solution is the hydrogen bond. So uncharged substances in aqueous solution, the solubility will be determined largely on their ability to produce or to form hydrogen bonds with water molecules. Another kind of weak interaction is van der Waals interaction, which is caused by the transient fluctuations of the electron clouds around an atom's nucleus. So the electron cloud is constantly fluctuating, and sometimes it happens that one cloud becomes a bit polar transiently, and it will stick to another cloud that becomes again a bit polar, only with a reverse charge. And these two species will attract each other up to the point of what is called the van der Waals balance point, which is the point in which the two opposite charges attract each other, and where this attraction is balanced by the repulsion between the two electron clouds. This is what we call the van der Waals contact point, or van der Waals balance point. It's an intermolecular, it's an interatomic or intermolecular interaction as of the intermolecular. It can be inside the same molecule actually. If the same base or say peptide, we get to know this term in a minute, but if a peptide maybe folds upon itself, then you have van der Waals interaction in one region of the same molecule with another region of the same molecule. So it can be intramolecular. It's an interaction between two atoms. The most transit one, sorry, yeah?
If there is nothing from the outside happening, will it stay that way, or is it so transient that
well, it is transient, it is transient, but it still has more energy than usually thermal energy, or the energy that we have here in the room. So van der Waals interaction would not break down spontaneously, but you do not have to have a lot of energy either to break them. So you can just hit the plate just a little bit, and the first to be broken are the van der Waals interactions. The minute they all, mostly the case is that you don't have just one interaction, but several ones, which succeed to stabilize a link between two molecules. So it's like if you have enough van der Waals interactions, that they will stay that way. They will stay stable. Okay, and the last interaction is more of an effect than an interaction, but still a very important one to biological systems. It is called the hydrophobic effect. Okay, so we saw what stabilized charged molecules in aqueous solution, and we saw what stabilized uncharged polar substances in aqueous solution, meaning the hydrogen bonds. But what happens if we have uncharged unpolar substances in aqueous solution? Then, okay, so this is an uncharged nonpolar substance, and what happens if you put it inside water is that the water molecules form sort of cages around it, pentagons and hexagons, and these cages have high degree of order, as you can judge just by looking at it, which means that they have low degree of disorder, which means they have low degree of entropy. They have low entropy. Now there is a law in thermodynamics that entropy strives to increase, and so this is an unfavorable, thermodynamically speaking, an unfavorable state. So if you take two such substances and join the surface of the cage together to form one cage, one bigger cage, the area of the surface in this case is less than this surface plus this surface, obviously. So you have less ordered surface and hence more entropy. So this is a favorable state, and it will stay this way. And the outcome is the two unpolar, uncharged substances are sticked together. Not because they have some glue between them, but simply because of the entropy that is gained or lost, or being lost around them because of the cages. And I think you know this phenomenon two wet glasses sometimes stick together in a manner that it is just impossible to break them apart. So what stands in the basis of that is the hydrophobic effect.
Is the water attracted to the molecules? If they are non-polar and non-charged,
Okay,
why do they stick around?
So maybe with van der Waals interaction, just a little bit, but they cannot form any hydrogen, neither ionic interaction, so you don't have any of these more stable weak interactions. So yes, the basic, what we think about hydrophobic effect, the fact that two unpolar molecules are being linked together in an aqueous solution, is this explanation, the entropy. I mean, this is the most accepted explanation.
I don't know if it's legal to ask for a translation during the course,
It's legal.
but can you try to explain the term entropy?
Entropy is just פשוט קוראים לזה entropy, entropia.
Okay.
You want to try to explain it in Hebrew?
No, or in English.
In English. Yeah, of course. It's legal. The cops won't. Okay. There's a long and exhaustive explanation in thermodynamics, but let's just stick with the definition that entropy is just a mess. The disorder, the degree of disorder of the system. So, for instance, a group of amino acids scattered around an aqueous solution are more disordered than if you take these amino acids and join them together to build a protein, because it restricts the manner they can rotate or translate or go anywhere they like. So you have less degrees of freedom actually. But you have less. Let's take it. Let's just say it like this: if the system looks more ordered, if the kid has cleaned his room, then the room has low entropy. And as you know, kids they tend to strive towards higher entropy. So they follow the law of thermodynamics. This was a bad explanation. I can try to do it again during the break as well. Okay, because less water is needed to form the cages, and we have increased entropy, a favorable thermodynamically situation. Okay, this is the relation between the different bonds we've just discussed along an energy scale. The units here are kilocalories. Calorie is just an energy unit. You can read about it. And a mole is what is called, again, just read about it at home if you don't know the term, is Avogadro number, which is simply a number. And in this context, it's just the number of bonds that you have to have this energy to break this number of bonds. That's what the units mean. So mole can be a number of chairs, number of atoms, number of dynes. But in this context, it is just the number of bonds. And you can read more about it. It's just some gigantic number, 10 to the power of 19 or something. Okay, so the strongest bond is the double covalent bond, for instance, between two carbons. And the single covalent bond is less stable. This is just one example that we will get to know later on in the cruise hydrolysis of ATP, just to put things in perspective. And then we have the much weaker interactions with them, hydrogen, electrostatic, and van der Waals interaction. And this is where the thermal energy, for the room temperature of 25 degrees Celsius. So again, as you asked about the van der Waals interactions, so they are the closest to the thermal energy. So they will not be broken spontaneously, but you don't have to have a lot of energy being and to put into your system to break them as well. Okay, when you talk about molecular complementarity, we actually talk about in a combination of part of all of these interactions we've discussed. So when two molecules encounter, they will most probably bounce apart from each other because of the thermal energy. And if they will have a recognition with the energy above that of thermal energy, taking into consideration all the different interactions between them, then they will stay linked. So you can see, okay, so this is an example of a weak global interaction between two molecules, and a stronger one, and you can see the ionic bond, hydrogen bond, van der Waals, and hydrophobic effect, and So this was atomic bond and molecular interactions. And now let's see what can we build with these different interactions, with these different bonds. So chemical building blocks of cells. Biological macrostructures, which also can be called polymers, are built from their monomers, from their building blocks. So, for instance, amino acid is the monomer of polypeptide, which is another name for a protein. Amino acid has a role as itself in the cell. It is, by the way, it can be a neurotransmitter. And nucleotide as its own also has a function. It can be an energy coin, so to speak, and we will see these terms as we go. But they also have a meaning when they are joined together to build polymers. So again, amino acids build polypeptides, which are proteins. Nucleotides build DNA and RNA, nucleic acids. And monosaccharides are joined together to build polysaccharides. At least one thing you don't have to dictate because it's just mono and poly. This is quite intuitive. And from each of these cases, we have a name for the bond that is being formed. So amino acids are joined together by a peptide bond, because it's a bond that exists in the peptide. Nucleotides are joined together to form RNA and DNA via the phosphodiester bond. This name describes the atoms that participate in it. A diester is through an oxygen bond. And the glycosidic bond stands in the base of monosaccharides turning into polysaccharides, because it's found in sugars, in glycosidic molecules. Note that each time the reaction, in the reaction we find an exit of a single water molecule. When we join the global principle in biology, when we join monomers to form polymers. Okay, these three bonds are covalent bonds. And there is also an example of hydrophobic effect that join monomers to polymers. So the monomer phospholipid, we will see its structure, I think, already today. So the monomer phospholipid is joined by the hydrophobic effect to build phospholipid bilayer, which is what the cell membrane is built from. And now let's go deeper into each of the examples I just noticed. So amino acids have a general structure of a central carbon atom surrounded by 4 moieties. Moieties is a functional chemical group. So I would sometimes maybe say moiety. It's like saying every chemical group in nature. Sorry? What did you say? Moiety? M-O-I-T-I? T-Y? I think it's pronounced moiety, or close to that. Okay, so also this is the basic structure of amino acids. We have an amide group, a carboxyl, and a hydrogen, and a side chain, which is also called a residual group, or just the R group for short. And this is the part by which different amino acids are different from each other. So, excluding the R group, this is the backbone of the amino acids, which every amino acid has. And with it, you have specific amino acids. It's actually, this is not a single atom. Obviously, it represents several atoms, which we will see what can be the combinations in. Okay, so have we just as we described, a tetrahedral can have 2 stereoisomeric configurations, and in amino acids we only find the so-called, never mind which exactly configuration is it, but we only find one of it, so-called L isomer. So this is the building block of proteins. And okay, we have 20 amino acids, and they can be classified by different measures of hydrophobicity, reactivity, structure, what have you. And we will use this then maybe the most common classification of according to their solubility or hydrophobicity. So hydrophilic is a substance that dissolves easily in water, because hydrophilic loves water, likes water, and hydrophobic is afraid of water, so it's not dissolved very easily. It's a tough solvent, I think it is called. So these 8 amino acids are the hydrophobic amino acids because they have no charge in physiological conditions. You can induce some charges upon them in experiments, but in your body these amino acids are, as you see it on the board, they have no charge in the residual group, which is the red part, the purple part in each amino acid.
The bottom one?
Sorry? All of them, all of these 8 are the hydrophobic amino acids. Because all of them do not have any charge across them. You see these have just carbon chains filled with hydrogen. These have these structures here are aromatic rings. Each vertex represents carbon. We just don't write it, but it has a carbon right here, and a hydrogen, again, we just don't write it. So it's some nomenclature if you like. And these hydrophobic amino acids usually can be found in the interior part of proteins, where they are hidden from the aqueous environment, because they are hydrophobic. Okay, each amino acid has its name, has its abbreviation. It's not a really abbreviation, but it has a 3-letter name and a 1-letter name, which you will, when you look at protein sequences in databases and so on, so you'll find the 1-letter nomenclature of amino acids.
They don't function in cells?
They do. They function in cells, yes, of course. What did I say that could mean that they not? So they function in cells. All 20 amino acids are found in cells. What I said is that they are, maybe that was the thing, they can be found in the cores of proteins, where they are hidden from the aqueous solution, because they have no ability to interact with the water molecules around them. Sorry?
They don't disassemble in the protein?
Other amino acids can be stable inside proteins thanks to interactions with other molecules, such as water molecules in the aqueous environment. But these ones,
What's special about them?
well, first they are hydrophobic, and functional speaking is that you'll find them in the core of proteins. So for instance, some globular proteins, a ball-shaped protein, you'll find these amino acids most usually in the interior.
I have a question.
Yeah,
The
sure.
way it's written, is it like for both D and L, or would I have to switch the H3N and the H2 for the isomers?
So first, remember that amino acids have only the L isomer in nature. I don't know why, but this is the case.
Okay.
But about the way we write it, which is the second part of your question. So when you write it like this, you don't see no configuration.
Okay.
So you have to, there's some convention of writing a triangular here, which means that this far edge is sticking out of the board, and so on. We will just use this conversion, so don't need it. Okay, the second group of amino acids is the hydrophilic amino acids, and they can be, okay, so this in opposed to the last group will be found usually on the surface of proteins, because they can interact and be stabilized with the water molecules around them. And they are hydrophilic for several reasons. The first subgroup is hydrophilic because it has a positive charge around it in physiological conditions. And there are 2 examples of amino acids negatively charged, which again interact with water, maybe oppositive with the hydrogen, but they would interact with water. And these 4 amino acids are uncharged, but polar, because these hydroxyl here and these amides, if it has, these amides are polar. So these 2 moieties can sustain hydrogen bonds with water, and hence would be stabilized, and hence is hydrophilic. Now, just note that we have a see the location of the aromatic ring here really do not allow for any hydrogen bonds or stable hydrogen bonds to happen around this moiety. Because I said that the hydroxyl is what gives is what stands in the base of the hydrophilic nature of the other group, but we do also have a hydroxyl here, but it collides sort of with the aromatic ring and hence hydrophobic. Okay, so these are the hydrophilic amino acids. We'll just finish the amino acid screen and then take our break. So this is the last slide before the break. 3 amino acids, special amino acids, should be looked at outside of this classification, hydrophilic, hydrophobic, because they are unique. So cysteine is unique in that it has a sulfate with a hydrogen is a sulfhydryl. It has a sulfhydryl moiety. Sulfhydryl, I think. Anyway, it has a SH moiety, which can, if encountered with another cysteine from the same peptide or from another peptide, right, from the same molecule from another molecule, but of another amino acid or another cysteine, it can produce a covalent bond between the 2 cysteines, and this is what we call a disulfide cross-link. A disulfide, because we have 2 sulfides, a disulfide cross-link. It's a cross-link because it's linked between 2 amino acids, between 2 regions of a protein or 2 proteins, and we'll see the disulfide cross-link in several examples, in the NMDA receptor, for instance, in 2 or 3 classes here.
What happens to the 2 hydrogens from the disulfide?
Okay, so while the cysteine or the disulfide link is being formed, you have an oxidation reaction in which the 2 hydrogens are exiting the system. This is like this. Yeah. That's what we call an oxidation. When you do it, you lose protein.
What's so special about this disulfide?
The special thing is that okay, so this cysteine can form a covalent bond. This is not true for the rest of the amino acids. This is what
You cannot obtain covalent bond?
other amino acids in the residual groups, no. They are covalently linked to the backbone of the protein or of the polypeptide right here to this region, to the backbone region. I mean, so we will see it in next class when we'll speak about protein structure and function, but basically the residual group cannot, maybe under extreme conditions, you can reduce other atoms as well to form covalent link, but cysteine disulfide crosslink, which is formed on cysteine, is a physiologically very common covalent link that is being formed in the cells. Okay, I would say like this. You can oxidize, under harsh environment, you can oxidize other atoms as well. Glycine is unique because as a residual group, it has a very full residual group, a residual group that includes only a single hydrogen. Question: would that molecule here would have 2 stereoisomers? Would glycine have 2 stereoisomers, 2 different configurations in space, which could not be overlapped? No, because you have to have 4 unidentical spheres surrounding the central carbon to have 2 different unable to overlap species of molecules. I'll try to bring a model next week, so we'll see exactly why it can be overlapped and can't be overlapped, because you just have to play with the 3D molecule to see why it is so. Okay, anyway, this you will a lot of times find it in tight spaces in proteins because it's so small. Just maybe it's pretty intuitive but also true. Okay, the proline is also a very special amino acid. Okay, so this may contrast what I've just said about the cysteine. This also has a covalent bond. The edge, okay, so you first have to visualize this amino acid without this bond here, just a chain of 3 carbons, and the edge of the chain can covalently be bonded to the amino acid, to the amide group in the amino acid backbone. So it circle around and interact very stably, covalently with the backbone itself. Only proline does it.
And will this bond stay when there is like protein formed?
Yeah, definitely. So the chain, the backbone of the protein can continue right here, and this structure will induce a kink, a rigid and stable kink inside the polypeptide. Yeah, so we'll see examples of that in the next week. So these are the three crazy amino acids. Okay. We will proceed with nucleic acid after a 10 to 15 minutes break. כן, כן. אבל בהפסקה. כי זה סובר. יש לי איזה כבר. כן, עכשיו יש הפסקה, התכוונתי אחרי. זה סובר. כן. קשה אבל בסביבה. אתה ממהר לעשות את זה בטח.
לא, נוכל לחכות.
אז זהו, כאילו, כבר.
אין בעיה. שבוע הבא אני אהיה במקלין. בגדול כאילו יש ספר.
אז יהיה מצגת, או דרך האתר או דרך מישהו ישלח את המצגת בשבוע הבא, אולי אני אשלח כן, תסתכל על המצגת. כן, לא כל הספר, דרך לעבור למחשב, אולי לראות את המצגת, ואז מונחים וסיפורים להגיע. זה הייתי מציע לעבור למצגת ולנסות לעבוד עם הספר או עם ויקיפדיה, וכל דבר שלא ברור, אז נתמשל.
המבחן עצמו הוא כאילו חזיתי ממש לדעת לפתח במולקולות, שמות שלהם.
אז זהו, כאילו, זה שאלה טובה. כי כאילו, אנחנו לומדים את זה ברף מסוים, ואז מה הרף שבו צריכים לדעת. הדרך הכי טובה לענות על זה היא פשוט, אז כאילו, as we go, נביאו את השאלות, אז תראה, לתאר לא, כי זה לא קורס בסיום כימיה, וזה לא בשביל אמריקאי בכלל. למה, נגיד לזה, מה אתה אומר?
זיהוי.
נגיד לזהור, אין הרבה אמיתי, לא, אין שם הרבה ביוכימיה מבנית. זאת אומרת, זה שיעור כמעט כולו על ביוכימיה מבנית, כי זה כאילו הראשון. כן, בדפי השאלות זה ייתן לך הרגשה של מה הסיפור בסוף במבחן, ואז כאילו, כן.
ויש באופן עקרוני מועד ב'?
כן, בטח, ברור. כמו כל, יש מועד ב' ואינטרמיון יש מועד ג', כמו כל החוקים.
לא, פשוט אני יודע שעלה לי את מערך מתחת למבחן במשקל מסוים. באופן עקרוני
כל אחד יש לו שני מועדים.
יש מבחנים של 30%, שזה בוודאות אפילו מועד ב'.
לא הבנתי, מה, לכל סטודנט, בכל קורס, בכל אוניברסיטה.
יש פסיכולוגיה, יש להם MA, תואר כאילו יש יותר מוזכר, ואז כל מבחן 30%, והם נותנים כאילו הם צריכים...
אני חושב נגד החוק שלנו.
לא, זה...
אם אתה מקבל...
אם יש מבחן שהוא פחות מ-30%,
מה זה הולכים?
משקל.
טוב, פה זה 40.
זהו, לא, אני לא יודע.
לא הכרתי נכון. אם זה בוחן. אם זה מבחן פחות מ-30%, זה מוגדר כבוחן.
אולי. כי אין מבחנים כאילו מעל ה-30% בקורס הזה.
תראה, המבחן סוף שלנו בשיעור האחרון, וזה מותר לי לעשות בגלל, דבר ראשון, כי זה יותר נוח לכולם, כי ברגע שנכנסים לתקופת מבחנים זה פחות נוח לכולם, אבל שנית, זה מותר לעשות חוקתית בגלל זה פחות ב-50%. זה מה שמישהו אמר. אני אסתבך פה משפטית. מותר, כן, בטח, יש מועד ב'.
אני פספסתי את הדקות הראשונות של השיעור, אז קצת זה, למדתי מדעי המוח במכלול הגדולים. צריכה, כי כאילו, אני חושבת שעכשיו פעם שיעור מהיר, אבל מבחינת הסילבוס, כי בכל היה לי קורס מומחים מדעי
תראי, זה הפרקים, זה השמות של הפרקים, בזה לא היית זאת אומרת. זה השמות
זהו, זה לא הייתי.
זה השמות של הפרקים של הקורס, השמות הגלובליים, זאת אומרת שאפשר להיכנס לכל אחד מהם הרי קורס בפני עצמו, השאלה כמה אנחנו ניכנס.
זהו, זה בעיקרון זה גם נדמה לי היה אותו ספר, אבל זה היה בשנה הראשונה של הספר, אבל כאילו, אני אוהבת לרענן דברים, אבל
כן.
השאלה היא איך אני יודע...
אני אעלה מצגות.
אוקיי.
נגיד 3 מצגות קדימה בקורס. ואז פה מגיעים נגיד חומר שהוא כאילו באמת נוירוביולוגי. ותסתכלי על המצגת נגיד האחרונה, תסתכלי אם הרמות של הדיבור היא רמה שאת מכירה או לא. את באה אינסטינקט? גם אם, אוקיי, אז אני לא יודע, אני אומר מה שאני הייתי בוחר בשבילך. כשתגיעי ל-PhD, אז תצטרכי לעשות עוד נקודות. אם יש לך אקסטרה נקודות מהמאסטר, תוכלי להעביר אותה ל-PhD. לעשות קורס שהוא טוב ותוכלי לקבל בו ציון גבוה, זה לא דבר. נתחיל מזה. נמשיך מזה, שבאמת, מצד שני לא צריך לעשות קורס שהוא מיותר ובטוח. זהו, בעצם לא הצעתי כלום, אז...
לא, אני מבינה, אני חשבתי על שני
זהו, שתי האפשרויות.
אני כן דווקא, גם אני באמת עבר עוד שנתיים מסתכלת אצלו, אבל בערך 5 שנים אחרונות, אבל...
אז אני מציע באמת את הדבר הזה, אני אעלה 2 מצגות קדימה.
אז אני אראה, אם זה באמת לגמרי ברמות שלא נראה לי, כי זה היה כאילו ביוכימיה, לאו דווקא בקונטקסט של נוירוביולוגיה.
כן, אז גם, כן, אז פה נגיד את המאמר.
זהו, אז איפה את הקונטקסט באמת שראיתי שקצת עוברים בטח את המאמר...
כן, נסתדל שכל דבר פה יש מאמר שכזה מיישם את הדבר בנוירוביולוגיה. אני חושב שזה, אז כאילו אני אשלח את המצגות קדימה ותעברי על עם הרמה הזאת של אם יש לך אותם.
אוקיי, אז אתה שלחת אותם לאימייל שלי ספציפית או שכי...
לא, אז אני אשלח, אז אני אעלה את כולם. אני אעלה את כולם אם יהיה אתר ואם באמת, אם לא אני אשלח אותם במייל, אין בעיה. אשלח את ה-PDF במייל. כן, אשלח את כולם.
תודה.
להשוות את המפורסם אתה חושב?
מה אתה אומר?
זו כבר מפורסמת אתה חושב?
לא, אני אשלח לכם אותה, בגלל שלא היה לי לא את הלינק ולא את האימייל ולא וובסייט עדיין, זה היה לאיפה. So first, all presentations, I will upload them and send them by email a week in advance before the class. This was an exception today because I didn't have your email and didn't have the website. And for those of you who have a biological background, so I would send maybe even today or tomorrow. I'll send 3 presentations in advance of the three classes of the 3 coming classes. And for those of you who have biological background and still don't know whether they should take the course or not, just take a look at the last maybe presentation that I'll send, and this one will already go deep into neurobiology, at least in the level which we will discuss in this course, and just see if you're familiar with the terms, if you're familiar with the phenomena that I'll describe, and if you are too familiar with them, then it's your call, but I suggest that you won't take the course, and if you're not, so take it. And I know there's some lack of courses for English speakers and in the neurobiology department, so everyone who's easy to read and dedicate the course is very welcome in our class, obviously. That's it. I'll send everything by mail, question page, and in the website. Question page, presentation, notes. Any questions, by the way, about administrative issues? Anything? Any requests? Go ahead.
What is your email? Just embed it in the...
Yeah, in the
okay.
first, but let me write it here for those who didn't catch it. It's long and bad, so prepare. I think this is A. Everything is A. gmail.com. That was the only thing that was empty. I'm adding 2 names. Okay, so nucleic acid. We continue with nucleic acid as an example, as another level of building blocks that comprise polymers. And as in the case of amino acids, and also here we have some general principle structure for nucleic acids, and then we have a component that differentiate between different nucleotides. So the global structure is a phosphate group, here in purple, attached via phosphoester. Again, a ester bond is the bond through via an oxygen, a steric bond, to a pentose or a ribose. A pentose because it has pentamer. Yes, pent meaning 5 carbon structure, and oz is the suffix. Yes, it's the suffix for sugar molecule. So this is why we call it pentose.
So this is about the nucleotide, the monomer.
This is one nucleotide. Yes, we're still describing the monomer. That's right. Sorry. So a phosphide linked via a phosphodiester bond to a pentose, which is a 5 carbon sugar molecule. Again, you see it's a language. When I say pentose, you should understand that it's a 5 carbon sugar molecule because of the pent and the O. And then a link via a nitrogen to a ring structure, which is also referred as a base. We call it the ring base. And this is in blue the part that is the variable part between different nucleotides. So this example, in here we have adenine, which is one of the nucleotides, and we still see the other 4. In RNA, in ribonucleic acid, the pentose is ribose, meaning that it has an the hydroxyl hydroxyl, sorry, in the second position, and in DNA, instead of a hydroxyl, we have a sole hydrogen, so it is deoxyribose, one oxygen less ribose. This is a very reactive. Sorry, what?
Two deoxyribose? Why two?
Okay, so no, again, no. The two was the position in which the hydroxyl becomes just is exchanged with a sole hydrogen. The hydroxyl is a relatively reactive moiety in the cell, so and this is the reason why RNA molecules are much less stable than DNA molecules. You know, you can leave DNA on the table and it will stay the same, pretty much stable. You can still extract its structure days, months, and even years afterwards. You know that we know the structure of mammoths found in Siberia recently and of dinosaurs, and we can't do the same for their RNA molecules. Which is the reason, by the way, which we will never be able to really, clone or create them from fresh, because not the whole information is stored in the DNA. Okay, I'm not sure about that, but never mind. So, but the hydroxyl here is the reason why DNA is so stable, because it doesn't have it. It has no hydroxyl in its pentose. It has in the other position, but this is the most exposed position, and it has no hydroxyl. Okay, so again, if it wasn't clear, these are the building blocks for both very important biological molecules, DNA and RNA. They're essentially the same. These are the 5 different nucleotides exist in nature, or the 5 different bases, right, exist in nature. We have adenine, guanine, and cytosine, which all these 3 can be found in DNA and RNA. Thymine is found only in DNA, and uracil is found only in RNA instead of the thymine.
What are the different components of the pyramid?
So a two-ring structure is called purine, and a one-ring structure is called pyrimidine. And it has a cool mnemonic, but just for Hebrew speakers. How would you remember that A and G are purines? Then you can think of Chag Purim Sameach as Ag Purim Sameach as Ag Purim Sameach. That's it. So what I just said is happy. How do you say Purim? Just Purim. Happy Purim holiday, and it gives a cool way to remember it. But you don't miss anything. That's the Hebrew. So okay. So these are the purines, and these 3 are the pyrimidines. In double-stranded DNA or RNA, the link is between a purine and a pyrimidine. Because this is the right distance between the 2 strands. A purine-purine link would be too long, and a pyrimidine-pyrimidine long would be too short in the double strand. Sometimes these deviations do occur, but they are very unstable. Okay, polysaccharides are another macrostructure of polymers that are built of some kind of building blocks, in this case monosaccharides. Monosaccharides are carbohydrate, which means that they are built of a chain of carbons with a 1-to-1 ratio with the water molecule. So on each carbon you'll find a hydroxyl and a hydrogen attached, which together form a hydrogen molecule. So we say that carbohydrates are a 1-to-1 ratio carbon-water molecule in different lengths, but always the same ratio. This example here is glucose, which is the monosaccharide with the highest availability of energy in nature. This is what we call the energy coin in cell. As an example, it can be found in the cell in different configurations, in a linear open configuration, or it can close upon itself to form two different circle structures. One is a hexagon, another pentamer, a pentagon and a hexagon, and this is just for glucose. But this principle, the idea that monosaccharides can form circular structures, are the same for any other monosaccharide. Okay, now because the link between monosaccharides is through the hydrogen then you can have, in principle, several ways in which two monosaccharides can be linked together to form a polysaccharide. Not only that, but also you can have more than one monosaccharide being linked to another monosaccharide. You can have two linked to a single monosaccharide, or even three, giving rise to sometimes branched structures of polymers, and not only linear structures of monosaccharides. So they are sometimes look like gigantic trees that branch. One branch is branching from the other, and so on, because of the hydroxyl groups, the multiple hydroxyl groups across each monosaccharide. These saccharides are the smallest polysaccharides exist. So let's see two examples of such molecules, and from them we will extract the principle of monosaccharide adhesion. Okay, so let's take the monosaccharide galactose and the monosaccharide glucose, allow them a 1-to-4 link through their hydroxyl groups, and you would get a lactose, which is a disaccharide, which is meaning there's already a polysaccharide. And for those of you who have trouble digest milk, then you probably know that this is the main sugar in milk. This is what does all the trouble. Take a glucose and allow it to attain a 1-to-2 configuration bond to fructose, and you will get the disaccharide sucrose, which is the main, not the main, but the main product, of plant photosynthesis.
Shachar?
Yep.
What are the numbers when you mention 1-to-4, 1-to-2? Is it the location, the position?
Yeah, it's the position between the two, because the hydroxyl, you have the hydroxyl in other exposed locations, then you can have other bonds giving rise to other polysaccharides. But the 1-to-4, 1-to-2 was the location of the carbon that is being attached to the other carbon, giving rise to a specific kind of polysaccharide. If, say, this carbon here would interact with another hydroxyl, say here, you will get another polysaccharide. Yes?
Where do you start counting these positions? Is it
Okay, from the oxygen and on, clockwise, and clockwise when you put the molecule with the biggest loyalty to the right, with the clock. But it's not that important. Nevertheless, I said 1-to-4, 1-to-2, just to note, just to give the idea, give the feeling that you can have different bonding configurations, or different localization of bonds, giving rise to different polymers. You don't have to know the convention of how you count sugar molecules. There's a certain convention around that. About the other polymers, amino acids, nucleic acid, we will discuss much more and way deeper in future classes. But about polysaccharides, this is pretty much the last time you will hear about that. So I'd like to say a few more words about them before we kind of neglect them. So polysaccharides function in the cell they have two major roles. The first is a reservoir of glucose, which is again the energy coin, the most available fuel exists in the cell, and they have structural roles as well. And here are a few examples. Glycogen is the most common storage carbohydrate in cells. Storage of what? Storage of glucose. So each tissue responsible for its own production of a glycogen storage, a process that is called glycogenesis. So we have glycogenesis in the kidney, in the muscle, and in the brain, which is highly important for brain function, and allows it to keep some degree of activity. And the primary storage carbohydrate in plant is starch, which is kind of the plant equivalent for glycogen. This is the main storage for the plant sugars. And another an example that fits more the structural component, the structural role, is cellulose, which is found in plant walls, cell walls, which is one of very flexible and yet very strong structure, and it is all thanks to cellulose, which is and that's what about the saccharide. Another basic molecule is the fatty acid, which is not a carbohydrate, but a hydrocarbon. And you only switch the order of these two words, and you get a different molecule. Hydrocarbon means a chain of carbons, not with a 1-to-1 ratio with water molecules, but not with any ratio at all, but just saturated or unsaturated, but linked across it with hydrogen. It has no hydroxyl molecule. And this way of writing is just to know that you can have several carbons attached to one another. In the middle, you would have 2 hydrogen attached to each carbon, because carbon can attain only and precisely 4 covalent bonds. And at the edge, you'll have 3 hydrogens for the same reason. And you can have different lengths of these molecules. About the nomenclature, you can have, so the first number here denotes the number of carbons in the carbon chain. How do you say nucleotide? Colon? First time ever. Okay, so to the left of the colon is the number of carbons in the chain, and to the right are the number of double bonds. So if you have no double bonds, if you have 0 on the right, so it means that you have the maximal number of hydrogen, meaning the molecule is saturated with what? It's saturated with hydrogen. So in the supermarket, when on the oil it's reading that it's unsaturated or saturated fatty acid, that's what it means. It means that it has the maximum number of hydrogen, or 1 less, or 2 less, or 4 less than maximal number of hydrogen in the molecule. And I don't say what is more healthy than or what's more tasty. But it is what we mean by saturated or unsaturated fatty acid. Okay, 2 stereoisomeric configuration can exist around the double bond. So first, let's say that in a saturated fatty acid, when you have no double bond, then there is a rotation around each single covalent bond, right? That's what we've already said. So on average, the molecule is linear. And this is not true when you have a double bond, which introduces a rigid, stable kink into the molecule. And this kink, theoretically, could be of two types, but empirically, it's only of one of them. You could have a cis configuration around the double bond, meaning if the 2 identical moieties are of the same side, of a line that is stretched parallel to the double bond. So these 2 moieties are identical and are on the same side of the double bond. The same for the hydrogen. Cis meaning on the same side, or close. You can find it also in cis Golgi compartment in the cell. We will know this compartment later on. And the second configuration is trans. Trans meaning on opposite sides. So again, stretch a line here, and 2 moieties are on opposite sides. And in the case of fatty acids, you only find a cis double bond. Cis double bond. So this is how the kink look like. Okay. And it has some significance, which we will enter later on. Yes. What was the question?
The last sentence.
The last, how many? all the paragraphs? Yeah, the last sentence about what so again, just as briefly introduce the idea of this slide. On a saturated fatty acid, you have a linear shape on average, because each of these bonds rotates around itself continuously. But on average, it's a linear molecule. And when you have a double bond, say here, you have a rigid kink. The double bond would be only of a cis configuration, meaning that the 2 carbons in 2 sides of the double bond would be of the same side compared to a parallel line being stretched right here between the 2 carbons that are engaged in double bond. And the trans configuration does not exist in fatty acids. I don't know why. Is it okay?
There is no trans fatty acid.
trans fatty acids. Right, it is written sometimes trans. I don't know, I check it. Maybe that's only for mammalian, maybe that's only for mammalian trans. And maybe in plants you do have trans. So it's not in the, I don't know the answer. I do know that this thing is made, I mean this principle written here, biological system contain only cis double bond. I don't think it's biological system. I think you're right.
Yeah, but I
I think
think
it's only mammalian trans. Yep.
but I think the trans are not in biological systems and they're therefore harmful in some way. They
So only when you cook it, the cis
it changes
to trans maybe.
Yeah, and then they think it's unhealthy because
Maybe. I don't know. I think the second most interesting subject after neuroscience is dietary studies. Really, I do think, but I never learned it. Maybe, I don't know. Okay, phospholipids. So, let's start like this. So we already know what fatty acids, what um, fatty, what fatty chains are. Take two fatty acid chains, stick them together via some esterification reaction right here, and then put a charged phosphate moiety and put some other, usually polar moiety, in this case choline, and you will get a very important molecule, the phospholipid. And what's so special about it is that it has a very hydrophobic area, which is marked by the yellow rectangle here, because fatty acids are not polar, are not charged, and have very weak hydrogen bonding capacities, so they're hydrophobic. And it has a very hydrophilic area, the blue rectangle here, which is hydrophobic because of all of this, you see all the charges that are distributed around it. And this feature, a single molecule that has also a hydrophobic area and a hydrophilic area, this feature is called amphipathy. Like this, so amphipathic phospholipids, that's how we call them. And it's a very, it's a single feature that has so much significance on the building of biomolecules and through that the building of electrochemical gradients across biomolecules, and through that the existence of spikes, okay? Because you want to have spikes if you don't have electrochemical gradients, if you don't have biomolecules, if you don't have amphipathy in phospholipids. And depending on intrinsic factors such as the length of the fatty acid, the identity of the polar head group, and so on, and also depending on extrinsic features, environmental features such as degree of saturation, means the concentration of how many phospholipids you have in your solution, temperature, pressure, and so on, you get different structures being built from the same building block, phospholipids. You can have mice cells, which are in which the phospholipids, their tails are facing towards the center. You can have liposome, where you have heads towards the outside and heads towards the inside, an inner sphere. And you can have a bilayer, a planar bilayer. And in the cell we find all such structures. Yes, so the bilayer is actually a fraction of the liposome, right? You scallop a piece of the liposome out and you have a bilayer. But still, in the textbook, this is the way they show it, because in the test tube you can produce this structure with exposed edges like this, like you look, I mean, like same as it looks right here. So they call it the distinct structure.
So the micelle and the liposome are both spheres?
Both spheres, but here you have an inner sphere. See the head, you have, this is a bilayer, this is, well, it's not really a monolayer, but yeah, you have an inner sphere, that's what differentiates between the two.
Amphipathic is with one hydrophilic residual and one hydrophobic.
Yeah, exactly at the same molecule, on the same molecule. You have two regions.
Two edges.
In two edges of the molecule, yeah. And two molecules with different hydrophobicity are not together called amphipathy, but only when you have it on a single molecule. was chemical building blocks of the cell. So we learned the basic interactions exist in the cell. We started to learn what we can build with them and introduce the notion of building blocks and macrostructures. Again, in the cell, the building blocks themselves, by themselves, have very important role. And they also build macrostructures, like a Lego. Okay, you can nature uses what it has, what it has. So it uses the macrostructures, and it uses the building blocks. Okay, you got the idea. A few words about chemical equilibrium. So before that, I guess most of you do know the basics of the chemical equation, but let's spend 2 minutes about that. So chemical equations are, let's write the simplest chemical equation that I know of. So hydroxyl plus hydrogen, these two are ions, cation and ions, gives under certain conditions can give a water molecule. So this is the chemical equation. In chemical equations, the left side are the reactants, and the right side are the products of the reactions. You have to have the same number of atoms on the same side, because material does not go anywhere. You have to have the same net charge in each side, so you have 1 minus 1 plus and a 0 charge, so everything is, only in this, if all these features exist, so you have a sensible chemical equation. And now, if I put the reactants in a closed vessel, and I let them start, the reaction start happen, then at the beginning you'll have, so the reactants will start be converted into products more and more, and at a certain point, you will have, the reaction will happen on the reverse direction. So most usually, and particularly in the cell, chemical reactions are not unidirectional, but rather bidirectional. They can happen both sides. And at a certain point, the products will start to be converted into reactants. So semantically, the products will turn into reactants, right? Okay, and then this is where the chemical equilibrium concept enters, and it is. Oops! No. Too late, huh? Oh, disaster. I pressed off on the projector. So, how much? You know how many?
You have to wait a minute.
How much? Never mind. We can do it without the. You know how much do we have to wait?
I think there's a light that when it maybe.
I don't have to press off twice for it to shut off. I only press it once. It has some, I don't know, backup mechanism. No, it doesn't. So, never mind. So, I'll try to turn it on while I'm. So let's turn on the light for now. Okay. So in chemical equilibrium, the rate of the two reactions equalize, the rate in which the reactants turn into products, and vice versa. The second these two rates equalize, this is where you have this is what we call chemical equilibrium. And just to clarify it even deeper, so it is not a step in which reactants stop being converted into products. And it is not a state where products are not converted into reactants. Not at all. Chemical equilibrium is when the rates themselves are equalized. So in this situation, you still have conversion between reactants and products and vice versa, but the rates are equal. So the concentration of the product stays the same, stays equal from 1 second back. And the concentration of the reactants stays the same. This is chemical equilibrium. Is that okay?
So again, it's talking about the fact that the content stays the same, or?
Yeah, yeah, the content or the amount or the concentration of the product stays the same. But it stays the same, not in a steady, well, it is a steady state, not in an unchanging manner. It continues to change, but the rate in which you gain more products equals to the rate you lose products. So you have no change in the concentration. Yep.
Does each product only have one set of reactants that it goes back to?
Well, under certain conditions, you can have, you can write different chemical equations for the same set of reactants, for example, but different, or sometimes you write it in one equation, but still it means that a certain set of reactants would give rise to a certain product or another one, and which would be built more rapidly or at all is determined by the conditions that you work in. By conditions, I mean temperature, pressure, moisture, and other conditions. Okay, and chemical equilibrium has a constant which connects between the concentration of the reactants of the products above and the reactants, and we call it the equilibrium constant. You have to raise each constant. Straight brackets denote concentration. So you have to range the concentration of each product or reactant in the power of its coefficient in the chemical equation, and you receive what you get is the chemical equilibrium. It is the same for each environmental condition. If you work at 1 atmosphere pressure and in 25 degrees Celsius, you will always, for a single reaction, for a given reaction, you will always have the same chemical equilibrium constant. You will reach equilibrium maybe more rapidly or more slowly, but you will reach it with the same ratio between the reactant and the product, which is represented by the chemical equilibrium. Say, for a given chemical equation, if you put a catalysator, it's in English?
Catalyst.
Catalyst, right. So if you put a catalyst in this reaction, you would reach equilibrium faster, but you will reach it, and then if you reach it already and you will calculate the chemical equilibrium, you will get the same number.
So the constant depends on the equation? Which equation?
What?
For each equation there is a constant?
For each equation, for each environmental condition, right? Because it will be it will be changed if you change the pressure, for instance. A different pressure will push you towards the reactants or vice versa. But for a given condition, if you know the chemical equilibrium, if all you know is the chemical equilibrium, and which you can Google, and you know the concentration of the reactants you put in a closed vessel, which is very reasonable to assume, you could calculate how many water you will how many products you will get. This is why the chemical equilibrium constant is good for us. Okay, it's important to know that in the test tube, when we write equations on the wall, so we think of the chemical reactions in the sense that reactants are being produced to products, and even go back and even backwards, but still we tend to forget that in the cell most of the chemical equations are not live in a vacuum. They are not distinct, but the products of one chemical equation serve as the reactants for another chemical equation. And so any calculation of the chemical equilibrium begins to be quite complicated in this sense. This is why we have pathway networks and such, and we try to understand how the different reactions are linked to one another. Okay, in the case of a non-covalent, so for instance, ionic interaction, generally of non-covalent binding, the equilibrium constant is called dissociation constant, KD, and this is actually the reciprocal of the chemical equilibrium for dissociation reaction. so because it's a redissociation reaction, so in the products we have the two separated, never mind, the two separated mers, and in the reactants we have the linked state. So just to see that it fits, that we call it dissociation reaction because the products are the dissociation products. And so if we have a relatively big dissociation constant, that means that we have more products. That means we have more dissociation. So in this sense, a small dissociation constant means high bonding, means less dissociation, more bonding, more stable bonding, binding.
Sorry, what do P and D represent?
P and D? P and D, so just two substances. In our case, it's just two substances that are unlinked. And they're just two substances that are unlinked. Let's just not complicate things. And here they are linked. So just I didn't add anything from what is written on the model. Here you have two distinct molecules, or you have one molecule.
Okay, so if KD is large, that means what?
That means that you have a lot of reactants. I mean, the reaction is more, it tends to be more towards this side, right? Monet, denominator, Monet, nominator. So the nominator is the product, I switched the, I should have written it opposite.
But the top is the reactant.
No, the top is the product.